Hey, what's going on, folks? Welcome to Science Grade 8. And actually, we're doing a double recording for this video, so actually, probably some of the 9th grader science students will be watching the same video. And if you're here this morning, I want you to say, what's up? Actually, it's not this morning, it's 11:45, but, say what's up, you guys. We're going to go over this test. You know, the test that I basically gave you guys the entire class last time to do in class. Well, I figured that you guys would have been done with it right by now, and so this is a Explore test. So you know that test, the test that you guys are going to be taking next week, the Explore test you guys are taking next week. Here's an old one. So this is an old science part of your test. So what I want you guys to do is just kind of think, okay, well, if after I go over it, if you find this easy, that's what your test is going to be like. All right? So it's kind of hopefully calming down your nerves so you don't freak out. All right, anyway, let's do this thing. I need to zoom in because it's hard to see, but it's still a little blurry, so I apologize. I'm just doing my best. I'm going to go through this. Let's go through question 1, and while I'm talking, you can ask your questions in the chat. Victoria, go for it. Ask your question. I'll read it and try to process it while I'm doing this. All right, you ready? Pull out your test if you have them. If not, just kind of watch what's going on, and we'll do this. Let's see. Why do we have to be at school for 3 hours if this test is only 30 minutes? Victoria, the reason why you have to be at the school for 3 hours and the test is only 30 minutes is because the science portion is only one part of the Explore test. There are 4 parts. There's a English, there's a math, there's a reading, and there's a science. And the science is the shortest of those tests. So I'm prepping you for the science part. Hopefully your math teacher is prepping you for the math test and whatnot. And actually, you know what? Here's the thing, is you guys are saying, oh my goodness, that's a long test, a 3-hour test. That's kind of standard when it comes to these kinds of tests. They're called standardized tests. I mean, it's kind of standard. And I think the biggest advantage of having you guys take this test is you'll get used to what Natalie is bringing up. She's talking about the SATs and the ACTs. These are tests that you're going to need to take in your junior and senior year. And really, I think that the thing that kills people on the test, and I'm teaching a class, I'm actually simultaneously teaching a class right now. I've got my juniors and seniors working on the actual ACT practice exam right now. They're doing a 3-hour day where they're just sitting in and taking practice tests because they got to take their ACT test next week. But I think that the thing that really kills people is that they just aren't ready for a 3-hour test. They don't have the stamina. So that's what I'm trying to build in you guys, is this ability to take these tests. Now, is the test the best way to measure how smart you are? No. This test really has no bearing on how smart you are. But this is the best solution that they could come up with, the colleges. So for the most part, even though I don't necessarily agree with having everything be standardized tests. I think that I can assess whether or not somebody understands what they're talking about when I talk to them and whatnot. Even though I don't agree with that, it's a necessary evil that I feel like I have to prepare you for. And it would be good for me as a teacher to prepare you for. With that said, let's turn to passage 1. We've got 6 of these to go through, so stop, stalling me for time. I need to get through all of this in an hour. All right, so my strategy for taking this test is this. For the science test, just read the questions. I'm not the questions, just read the passage. Start off by reading the passage. Don't worry, and here's my problem, is I get so worried in trying to figure out like every single thing about the thing, and they're saying, okay, you're going to build this circuit, and then we have this many resistors and capacitors, and I'm trying to figure out the science, and I look at it, and I'm reading it. As I read it, I try to read it as if it's a textbook and all of that kind of stuff, and then the question asks me something like, what is the x-axis of the graph? And I was like, I didn't need to know anything about that experiment to tell you that information. So don't slow yourself down with trying to understand every single thing that they're saying. Just read it. If there's a graph, Take 2-3 seconds to look at it, understand what the parts of the graph are, and then move on and then answer the questions. And you'll find that this test is mind-blowingly cool. All right, you ready? I'm going to read it to you. Ready? Scientists studied how conditions at Lake A change as the depth of the water changes. All right, cool. They collected samples of lake water at the lake surface and at 1 meter intervals below the lake's surface. For each sample, the scientists determined that the concentration of phytoplankton, what are phytoplankton? Well, they'll tell you. They're microscopic organisms that use sunlight to make food. Okay, they're plants. Got it. And the concentration of dissolved oxygen. See figures 1 and 2. Okay, so this one's phytoplankton. Let me just kind of do this and then do one of these and then give me a second. All right, phytoplankton, dissolved oxygen. Got it. The scientists measured at 1 meter intervals below the lake surface and the percent of sunlight that penetrated the water. So here's the sunlight. That's figure 3. And look, 1 meter intervals, so that means they went at the surface and they measured the phytoplankton. Any phytoplankton over here? Nope. Okay. They go down 1 meter. Any phytoplankton under here? Yeah, no, not really. Any phytoplankton over here? And they go down all the way to 5 meters. And they're like, oh, all the phytoplanktons are hanging out over here. And then they go down and down and down and down. Cool? There. That's pretty much all you need to know about the problem. Let's go to the questions. You guys got it? Now, I'm sorry that it's a little laggy. Make sure you let me know if you can't understand what I'm talking about. All right, okay, ready? According to figure 1, all right, let's look at figure 1 again. All right, the Lake A water sample collected between the following depths is most likely to contain the greatest concentration of phytoplankton. 3 to 4 meters, 5 to 6 meters, 7 to 8 meters, and 9 to 10 meters. I'm going to view, zoom out, and we'll take a look. Here's figure 1. Look, highest concentration of phytoplankton looks to me like right around 5 and 6, so I'll just say B, 5 and 6. Are we cool? Boom. Yep, that's it. See, I didn't need to know anything about how phytoplankton is created or all of that kind of stuff. I just need to look at the graph and say, oh, interesting. All right, can you guys see this if I read it from here? I know it's small. I apologize. I can't get like a middle version of it. All right, okay. I'll read it to you, and you guys can kind of just trust that I'm reading to you guys the right thing. All right, I'll read it to you, you listen. How's that? A scientist claimed that there was not enough sunlight for the phytoplankton to survive at 12 meters. Does figure 3 support this? Okay. Well, look at 12 meters. And looking at figure 3. And figure 3 is measuring what? I'll zoom in if you can't see. Sunlight. Yes. How much sunlight can penetrate 12 feet of water? 0. So, by the way, also look at how much phytoplankton are at 12 feet of water. I know you guys are already answering it, but I mean, I'm just saying. There's none. There's no phytoplankton at 12 feet of water. There's also no sunlight at 12 feet of water. Looking at that, I would say that, does figure 3 support my claim? I would say yes, it does support my claim because the sunlight penetration is 0. So the answer is J. Take that, folks. Does that make sense? So I'm reading this and it says, some scientists said that not enough sunlight for phytoplankton can survive at 12 meters. I looked at the thing and it says, oh, okay, there's no sunlight at 12 meters. That's why. Got it. All right. According to figure 3, the greatest change for sunlight penetration occurred over which of the following ranges? Okay, let's look at figure 3. Here's figure 3. It went from 100 down to 30 in 1 foot. It went from 30 to 20 in another foot, from 1 foot to 2 feet. I mean, looking at this right here, where's the biggest jump that you see in the graph? Yeah, exactly, between the first foot, 0 to 1. So that answer is 0 to 1. I mean, come on, you guys, challenge me. All right, ready? According to figure 2 and 3, the lake water has the dissolved oxygen concentration of 8.5 milligrams per liter and a sunlight of 23% at what depth? Okay, let's look at it. So, which one gives you 8.5 milligrams per liter of dissolved oxygen? It would be any point that lies on here. Got it? And then which one gives you 23%? Well, this is the sunlight penetration percentage. So 23% lies right here. Agreed? Therefore, if I were a betting man, I would say that your answer is got to be around 2 feet or 2 meters. One of those, yeah. Cool. So the answer is actually, by the way, on this test that you're looking at right here, it's F. But when you put it into the answer key, I actually had you put it in as A, because I didn't make a B, C, D, F, G, H. I did a A, B, C, D, all of that kind of stuff. So whatever. But you guys understand the question? How that one does? Done with passage 1. It's not that bad. What do you guys think? Was that like something really hard, or like you really needed to know everything there is to know about phytoplankton? No, not really. No, it wasn't that bad at all. All right. So don't stress yourself out. Okay. 2 studies were done. All right, passage number 2, part number 2. You guys ready for this? 2 studies were done in a lab to investigate the amount of carbon dioxide, in the volume of gas, or in a volume of gas affects the rate at which the temperature of the gas increases. That's a doozy of a sentence. Read that one for yourselves and see if you guys can figure that out. 2 identical glass tanks were airtight with airtight lids, A and B, are open, and the same quantity of black sand was added to each. All right? So I have, let's do this. Let's make tank A, tank B. They're the same size. And they have black sand inside of them. Fine. Black sand, black sand. And I added the same exact amount of sand to each of them. Here's A, here's B. We cool? All right, moving on. The heat of the lamp is 150 watt light bulb. Got it. 150 watt light bulb. 150, 150. Same light bulb, same everything. And placed in the open, the thermometer was set up so that the bottom of the thermometer would be 5 centimeters above the top of the sand. Got it. By the way, just to let you know, I needed none of that information. I didn't even need that picture. But I'm letting you guys know. See, look, they drew the picture for me right there. All right. Study number 1. Here's what they did. The lid of the tank was closed. Oh, yeah, I'm recording on a different computer. Okay. The lid of the tank was closed and the sand was added, or after the sand was added, 5 minutes later, the temperature of the tank was recorded. Immediately after the lamp was turned on, and the temperature in degrees Celsius was recorded every 30 seconds. And that's figure 1. Okay, all right. So it looks to me like they put the sand in, they waited 5 minutes, and then they started recording this. And every 30 seconds, they recorded, you know, it looks like it's going up. Got it? Got it. Okay. So what was inside there? There was air inside of there. So let's make a little air inside there. This is tank A. They put air in there. All right. Study 2. The lid of the tank B was closed after the sand was added, just like in Study 1. And through a tiny hole made in the lid, all the gas that was present was removed from the tank and replaced with CO2. So it's a different gas. 5 minutes later, the temperature of the gas in the tank was recorded. Immediately afterward, the lamp was turned on, and the temperature of the lamp was recorded every 30 seconds for a 10-minute period of time. The results shown in Figure 2. All right, what is the difference between Experiment A and Experiment B? I mean, you can almost look at the picture and say, oh, that's it. What is the difference that you see? The gases. Is the lamps different? Is the amount of sand different? What about the height of the thermometer? No. It's all it is we just replaced it with one had air and one had CO2. That's it. That's the difference. I mean, if you experimentally want to try to check something out, those two, I mean, you don't want to switch every kind of variable. You just want to switch one thing to see if the CO2. Got it. All right. Let's look at the picture, and that's what it looks like. All right. When you look at a graph, maybe it might be interesting to see, well, how high does it go? It goes to about 30. Let's look at the other graph, by the way. This is when it only had air inside. How high did it go? About, no, not 15. I would say, what is that, about 27 and a half? It looks like to me 27 and a half. We're all in agreement there? Okay, here, let me zoom out a little, or let's read the questions. According to Study 1, the temperature of the gas at 0 minutes was what? Oh, seriously? All you do is read that. All right, at 0 minutes, the temperature was 23 and a half. Everyone agree? Boom. I mean, it's like I didn't even need to know anything about like CO2 and how it, it affects the temperature and all that kind of stuff. They're actually asking you to read graphs here. According to studies 1 and 2, the temperature of the gas at tank A at 10 minutes was nearly the same temperature as the temperature of the gas at tank B. Okay, the temperature of the gas in tank A at 10 minutes was 27.5 degrees Celsius, right? Degrees Celsius. Let's look at tank B. At what time are you going to hit a temperature of 27.5? 5 minutes. There's your answer. We cool? Do you mind if I zoom out? I just like seeing the whole thing on one page. All right, next problem. The temperature of the gas in which the tank increased more quickly more quickly, and why? So which one increased quicker? Tank A or tank B? Raven says tank A. Raven is 100% wrong. I'm sorry, Raven. It's not tank A. I mean, if you look at it, this one only goes up to 27.5 degrees. This one goes up to 30 degrees Celsius in the same amount of time. So tank A is not the right answer. So you can kind of cross out both of those options. Got it? Okay, this is B and this is A. Cool? All right, tank B. Yes, got it. Okay, so which of these answers is correct? Tank B, because the gas of the tank, gas in that tank contained less CO2 than the gas in tank A. No, that's not true. Tank B had more CO2, so the answer is D. There we go. That's it. It didn't even ask me like that. I mean, yeah, I don't even know how to make that easier. It's pretty straightforward. All right, suppose that what occurred in tanks A and B after the lamps were turned on were intended to model how the temperature of the Earth atmosphere changed in the amount of time of as the amount of CO2 in the air changes. What would have been represented by the black sand, and what would have been represented by the lamp? Okay, so we're talking about how this could represent how the Earth acts. And your lamp should represent some kind of energy source. What do you think? The lamp is the sun. I totally agree. What about the black sand? What do you think that is? It's the Earth, exactly. So the black sand is the Earth's surface. The lamp is the sun. There it is. Answer G. We coolios with that, folks? Ta-da! Yes. All right. In studies 1 and 2 together, where studies 1 and 2 together designed to determine the brightness of the light received by the volume of gas affects the rate at which the temperature of the gas increases. Wait. They're saying, they're saying, for study 1 and study 2, was I measuring how the brightness of the lamp changes your temperature? No. I didn't change the lamp. I kept the same lamp. So both of these options are out because I didn't even change the lamp. How can I measure the difference in brightness if it had the same brightness? So the answer is no. And why no? Because the brightness of the light received the same on both. B. There we go. You guys ready? Can I move on? Boom. That's right. All right. I skipped this problem. I'm not sure why I didn't give that to you in your test, but sorry, you didn't get that one. So we'll skip it in our answer thing because we're running out of time. I hate to say it. I love hanging out with you guys, and I always run out of time. That's just the nature of this beast. All right, let's do this. Ready? Oh, gosh, this one was a weird one. Do you guys remember reading this one? Now, this is why I don't like teaching biology. In fact, I've never taught biology. Thank goodness, because you would get weird stuff from me. Anyway, let's try to read this as best as possible. This is passage number 4, and it's talking about like a fruit called pyrifolio. All right. Anyway, why don't we just kind of read through it, do our best, and answer the questions? Because I don't understand everything that's going on here. I'll read it to you. Ready? Fruit size in a pear, P. pyrifolio, is determined by the number of cells that grow in the mesocarp. What's the mesocarp? It's the part of the fruit that you can eat. During the period of cells division, what is that? Well, it's the average number of days of active cell division during the pollination. Okay, all right, I don't really understand what's going on there, but Table 1 lists fruit size at harvest, maturation period. What is the maturation period? The average number of days from pollination to harvest. Period of cell division and average mass of the fresh fruit of harvest of 9 strains of P. pyrifolia. The P. pyrifolia is so weird for me. Can I just change it to papaya? Can I change that word to papaya? It helps me. I'll just think about it as papaya. Okay, so apparently we're dealing with papaya. That we have big papayas, hence the large, and we have small, medium papayas and small papayas. And apparently those are different strains of the same kind of fruit. So they're like different types of papaya. You know, you got like green papaya, you got like yellow papaya, you got chicken papaya, all of that kind of stuff. It's just, you know, that kind of stuff. All right. So what is this graph right here? This is the average number of cells in the mesocarp sample is shown in each of the 3 fruit sizes. See figure 1. All right. So the average number of cells at pollination. So apparently that's the beginning of their life, because when you pollinate something, you know, when we talked about the birds and the bees and how the bees would pollinate the flowers. Actually, I have no idea why it's birds and bees, because bees don't pollinate birds, but yeah, anyway. Yeah, we're talking about pollination, and that's the beginning of their cell division and all of that kind of stuff. It looks to me that like those fruits are about the same size when they pollinate. All right, and then when they're harvested, that means when you can pick them off and eat them, obviously the big ones have more cells and the small ones have less cells. Got it? All right, so let's read the table, because I have a question. Are you experts on this passage? Do you understand what's going on? Because I'm not. In fact, I talked to an expert on this passage, and they were just as confused by this question as I was. But why don't we answer the questions anyway? Let's just do it. I mean, whether or not we're experts, let's just do it. I mean, I hate to say it, but I mean, man up and do it, even though you guys aren't all men. But, all right, let's do it. All right, forget I said that. According to Table 1, on average, the fruit which papaya strand, or a fruit of which papaya strand will spend the least amount of time in the tree from pollination to harvest. Okay, so the amount of time between pollination and harvest is called the maturation period, right? Because they told me that that is the average number of days from pollination to harvest. Got it. So which one of these spend the least amount of time? Well, this guy only spends 105 days. There's your answer. Boom. I don't know nothing about what they're talking about, but I know the answer to that question. I love this kind of test. All right. Based on Table 1, if pea papaya fruit which found to have a fresh mass of 810 grams at harvest, which category would it most likely be sized as? Well, let's see. This is the amount of grams that we're talking about. 810 falls right about here. That's still a large fruit, right? Because you can get all the way down to 765 grams and it'd still be a large fruit. So these options are out. And then let's read this. They say it's large because 810 falls between the average mass of S1 and S3. Yeah, that's exactly it. There's S1 and S3. 810 is between that. It's large because the average mass falls between S7 and S9. That's totally not true at all. So the answer is C. All right, you ready? According to Table 1 and Figure 2, to the papaya strains with the longest period of cell division have the greatest number of cells in the mesocarp at harvest. Don't really understand what they're saying with that, but I think that if I read that, I kind of say, all right, these guys spend a long time. These guys don't spend a long time. These are big because they have plenty cells. These are not big because they don't have plenty cells. So therefore, the answer is yes. I think that the ones that spend the most time between pollination and harvest are the ones that, yes, I'm sorry, Victoria, yes, you can go. I'm sorry that you're having tech issues. I wish you didn't. Everybody else let me know if you have tech issues. Let me finish my thoughts on this. I would say that the answer is yes, so it's not these. Because, I don't know, that's what the graph says. Let's see. What are the options? Yes, because the large fruits have a greater number of cells in the mesocarp than the... Yeah, that's obvious. All right. Yes, because the small fruits have a greater number of cells. That's obviously not true. You guys get it? Was that hard? No, not really. I didn't even understand what's going on, really. I just kind of answered the question. I mean, I love this, the fact that I am not a biology guy. I actually went over and told the biology teacher that I will never want to teach your class. And she said, good, because I hated teaching physics. And I was like, physics is so cool. And then we got into a fight. So Mrs. Sononini, the biology science teacher, and me, we were arguing about which one's cooler. And I actually think that, I don't understand biology. And here's what I mean by that, because, and by the way, people from Ms. Sononini's class are going to be watching this archive, so that's right. I said it. I don't understand your crazy science, you. But, well, let's move on. Let's answer question 17, and here's a story that I'm going to tell about that in just a second. Which of the figures, figure 1 and figure 2, if either, shows for papaya the average number of cells in a mesocarp sample before the period of cell division? Okay, here's the problem, all right? Ms. Sononini is a brilliant person. I'm not being sarcastic about that. I think that she's brilliant because she understands biology. And biology is so complex to me. Like, I don't understand what's going on. So, she was arguing, oh, this is such a hard question. It could be figure 1, it could be figure 2, because technically there are sex cells that you know, like have multiplied and pollinated, cell divided over here, and so maybe this might show it, and then this might show it. And the way I approach this, you guys want to know how I approach this? I said this. I said, I don't understand anything about this, but what I do understand is that this is when it pollinates and this is when it cell divides. And it looks to me that most of the cell division happens between when it pollinates and cell divides. You guys agree? I mean, look, you have 15 cells over here, and over here you have 800 cells. Looks to me like most of the cell division is happening between pollination and harvest. Agreed? So therefore, they're asking for which one shows before the period of cell division, and I don't see anything of that in the graph. So I'm going to go with that answer. And you know what the funniest thing was? Ms. Sononini had to agree with me. Because we're assuming that you are not biology teachers and you know all about how, you know, the cell division actually technically happens over here, so technically your answer could be figure 1 and all of that kind of stuff. This test doesn't assume that. I have no idea what I'm talking about. I'll be honest with you. Passage number 4 just kind of eludes me. I can get the answers, and I can get all of the right answers, and that's my point, is that you don't need to know everything there is to know about how to solve these things. You just need to do it. If I take this test and I get all the right answers, and Ms. Sononini takes the test and she actually understands it and gets all the right answers, we're going to get the same score. You know, my score out of this one will probably be a 4 out of 4, and hers will be a 4 out of 4. And they're not going to write a note and say, by the way, he didn't know what genome, I don't even know if that's a word, the pyrifolia is, and what mesocarp means, and all of that kind of stuff. By the way, he didn't know all of that. They're not going to do that. They're just going to, I don't even know what I'm talking about in that regard. But I got all the right answers. She got all the right answers. We'll both get perfect scores on that. But I mean, see, that's my point that I'm trying to make is I don't know anything about biology. You guys probably know more about biology than I do, and I'm a science teacher. I'm actually a good science teacher too. And I can still pass this test. So don't freak out if you don't know everything there is to know. You guys see what I mean? Like, for example, this question that I skipped, I have no idea why I skipped it, because that's a physics question. In fact, this one has a lot to do with that skateboard theme park that I made you guys do. Remember we're talking about gravitational potential energy and kinetic energy and all of that kind of stuff? You know, maybe Miss Sononini knows nothing about physics in that regard, but I guarantee you that she can answer these questions because they're kind of straightforward. All right, anyway. Let's go over these last 2 problems in 10 minutes, and then hopefully I'll be done, because I have to go. And I'm sorry if I go over in class extra time and all of that kind of stuff, but you know me. I like hanging out with you guys. I actually really enjoy teaching this class. Let's move on. Tooth decay is caused by bacteria in the mouth, such as and mutants, and S mutants. Got it. All right. I'm going to call one cavities and mutants. Let's call it mutants. How's that? All right. That turns sugars into acid and dissolves tooth enamel. 2 studies involving these bacteria were performed. 5 students, study 1. 5 students chewed sterile rubber bands for 5 minutes without swallowing. Depositing their saliva in separate sterile containers, they added 20 milliliters of sterile test agar. Got it? And the TA, which is the test agar, contains sugar and other nutrients and the growth bacteria, blah, 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 blah. If the amount of, and what happens with this Lactobacillia is it turns green if it's good and yellow if it's bad. I'm paraphrasing at this point. I hate to say it. I'm sorry. In addition, they added sterile water of blah, blah, blah to 0.2 milliliters of each student's saliva in each tube. Finally, they incubated the tubes at 37 degrees Celsius and noting the color of the TA at 24, 48, and 72 hours. Got it. So they basically waited a day, and then the next day they measured, the next day they measured. I'm good. The higher the concentration of blah, 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 blah in the student's saliva, the more quickly the TA changed from green to yellow. So it looks to me that like if you've got a funky mouth, it's gonna, this test tube is gonna turn yellow. And if you like keep your mouth clean, like brush your teeth more than once a week, then this test tube will stay green. Well, hopefully you brush your teeth more than once a week. All right. So it looks like sterile water, so pure water, stayed all G. That's a good thing. Pure bacteria turn yellow immediately and just kind of like stay it all the way down. All right. Student A obviously does not brush his teeth more than once a week. Student B brushes his teeth and then like, yeah, I guess he brushes teeth, but maybe not that great. Student C is like the kid of a dentist. Student D looks to me like, you know, I mean, they're doing better. Maybe the kid of a person who has a relative as a dentist. And then, you know, student E is some kid who brushes his teeth often as well. All right, got it. The students tested the effects of baking soda and hydrogen peroxide on the growth of mutants. They set up 4 growth plates and then added to each of the plates 2 or more substances listed on table 2. Finally, they incubated the plates at blah, 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 and counting the number of plates for 5 hours and 30 hours. By the way, I'm actually very sloppily going through this stuff, reading this stuff, just to kind of show you that I'm skimming. Got it? And it looks to me like they're trying to make toothpaste. So, in this toothpaste, they put sugar, mutants, baking soda, and water. Or that's not water, that's hydrogen peroxide. And hydrogen peroxide. Actually, exactly, yeah. They say it right there, hydrogen peroxide. Got it? All right. In X, they put in sugar and mutants. In Y, they put in sugar, mutants, and baking soda. In Z, they put in sugar, mutants, and just hydrogen peroxide. Got it. Okay. And here's the number of bacteria for each of these plates. So it looks to me like your W and your W and your X plate, they kind of suck as far as toothpaste because it's got a lot of bacteria. Whereas these guys are good inside of toothpaste. Okay, fine, let's answer the questions. I kind of don't understand what's going on. Let's just answer the questions. All right. According to Table 2, in which of the following ways did plate X and Y differ in regard to what was added to them? Oh, okay. So we have plate X and plate Y. Look at these guys. What's the difference between X and Y? No, I mean, don't give up like that. I mean, look. In X, I have sugar, mutants, and no baking soda. In Y, I have sugar, mutants, and baking soda. So the answer is the difference is baking soda. So the answer is F. Because F says baking soda was not added to X and added to Y. That's it. That's your answer. Does that make sense? I mean, it wasn't even that hard of a question. All right, you ready? Based on the study 2, mouthwash with which of the following ingredients would best prevent tooth decay? Okay, let's see. It looks to me like W and X kind of was junk in preventing tooth decay. Wait, based on the results of study 2, mouthwash with which of the following ingredients would best prevent tooth decay? Oh, okay. I think it's B, to be honest with you. Because B, like, they all had the S mutants in there. You know what I mean? Let me think about this one. Mouthwash. I need to think about this one. Give me a second. Hold on. Let me think. Based on study 2, mouthwash with which of the following ingredients would best prevent tooth decay? Okay. So it looks to me, I'm going to analyze this because I think that when I wrote this down, I was discussing with the science teacher, I think we both got it wrong. Let me re-look at this. All right. So it looks to me like baking soda was good, right? Because Y is down here. I can't even see it very well. Let me zoom in a little bit. In fact, I'll look at the paper. Oh, I see. Okay. So this is plate Z down here. This is Z. And this is Y. And this is W. And this is X. So Y and Z were good. Good. W and X, junk. Got it. Okay. So what did Y and Z have that W and X didn't have? Yeah, I say baking soda. Because, well, I would say that the answer is baking soda or hydrogen peroxide. But there is no hydrogen peroxide answer here. So I would say that it's just baking soda. If they had, you know, A, sugar, B, hydrogen peroxide, C, baking soda, or option D, you know, both sugar and or baking soda and hydrogen peroxide, I would say that that's the answer, but apparently it's not. So I got C. And somebody told me that I got it wrong. I don't think I got that wrong. I think you misread the question, Sononini. Ha. All right, let's move on. I got 3 more questions to cover. I've got 1 minute to do it in. Let's do it. In study 1, why did the students choose sterile rubber bands? Did that have anything to really do with the experiment? I think all it did is it made them get spit in their mouth. Done. In study 1, the student whose saliva contained the lowest concentration of lighter bacteria was the one whose one was, yeah, it was green. Yellow is bad. It was green all the way. So option C. I'm not even going to look at the graph. Boom. All right. In study 1, which of the following was most likely the reason the containers used to collect were needed to be sterile? Well, because when you do an experiment, you don't want like bacteria inside of your containers before you do the experiment because you don't know whether or not the bacteria was caused by the spit or it was caused by the stuff that was already in there. Cool? Now, I didn't finish this passage over here. I didn't finish 2 out of these 6 passages, and I talked a lot about this stuff. And I apologize, and I'm going over time, so I'm done recording this class. Have fun, folks.